Sanger sequencing revolutionized the area of human genetics and genomics. It is indeed a boon to the human race. Hello everyone, welcome to the online lecture on Sanger sequencing. Myself, Dr. Arul Jothi from SRM Institute of Science and Technology, Chennai. In this lecture, we will be covering the principles and key concepts of Sanger sequencing, and also we'll go through the workflow of Sanger sequencing. Sanger sequencing technique was developed by Frederick Sanger in 1977. This technique involves a selective incorporation of chain-terminating dideoxynucleotides during the DNA replication. This would result in fragments of different size which can be separated based on its size to reveal the sequence. We will be covering some of the key concepts on how this Sanger sequencing works. Chain termination. Before understanding this concept, we have to know what is happening with the DNA polymerization. In DNA polymerization, the 3' hydroxyl group attaches with the subsequent 5' triphosphate to form the phosphodiester bond. You can see here the hydroxyl group of the existing chain attaches with the triphosphate of the new nucleotide. So this hydroxyl group is very crucial for adding the additional or extra nucleotide. Whereas in case of dideoxynucleotide, we have an oxygen removed at the second position, also an oxygen removed at the third prime position. So that's why we call it as dideoxynucleotide. In this case, there is no favorable condition for this triphosphate to form the phosphodiester bond. So whenever there is a attachment of dideoxynucleotide, there will be a chain termination. So if you see in Sanger sequencing, the DNA fragment or a PCR product is being replicated in presence of normal deoxynucleotides to enable a normal DNA polymerization. Plus, there will be small amounts of chain terminating dideoxynucleotides added. So there will be a normal strands in place. Also, some strands whichever incorporates these chain terminating dideoxynucleotides, they will be halted. Okay, so these dideoxynucleotides halting the DNA synthesis at specific positions will result in a series of fragments that will differ in size by one nucleotide. So these fragments are then separated by this size to determine the sequence. Floresent labeling is the next concept where these dideoxynucleotides are labeled with unique fluorescent label. For example, A nucleotide is labeled with the green fluorescent dye, T with the red, and C with the blue, and G with the gray dyes. So these fluorescent dideoxynucleotide tag fluorescent label serves as a signal molecule to represent which nucleotide is at the end position. You remember when the DNA is getting polymerized in the presence of these dideoxynucleotides, there will be a chain termination, and upon which there will be different sized fragments produced in the reaction mix. So each size will be ending with one specific nucleotide, which can be identified with this fluorescent labeling. Back then, when people used conventional Sanger sequencing, this radio labeling was not there, but this radio labeling is an advanced form of Sanger sequencing, where the radio labeling, where the fluorescent labeling gives uh, uh, allows for automated sequencing. So, following the cycles, the fragments that are produced, the different sized fragments that are produced in the cycle, are made to run through these uh, electrophoresis, capillary electrophoresis, where the fragments of different sizes will run through it, and the smallest fragment, which we consider the first chain, after they will be having primer, say for 20 base pairs, and the fragment with 21 size, 21 base pair will be read first. The fragment with 22 base pairs read second. the fragment with 23 base pair, right there. It keeps on going in the same way. So we can consider the sequence from the 21st base pair. Okay, so we have attached different fluorescent labels. So A has got green, and T has got red, and C has got blue, and G has got gray. So based on the chain terminating nucleotide, the signal will be recorded here. If the A passes through, clever chromatogram of green, and if T passes, the chromatogram will be of red, and C, the chromatogram will be for of blue color, and if there is a G, it will be of gray color. So that's how as the nucleotides, I mean, as the DNA fragments of different size passes through this, the fluorescent molecules signals are recorded here. And that's how we come to know the sequence in the same order of the size. Now we will see how this Sanger sequencing work flows. So basically now we have to get a DNA and use gene specific primer, fragment specific primer, and we should give the PCR product. After that, the Sanger sequencing work flows like the cycle sequencing like this, and that will be followed by capillary electrophoresis, and which will be followed by the reduction of signal by the sequencer. Yes, this is the pictorial representation of Sanger sequencing. So while doing this cycling sequencing, you can see that this is the primer, and these are the chain terminating nucleotides. As you can see, there are different fragments differed by one nucleotide. So this one has got primer plus one nucleotide, the first nucleotide, and this has got primer plus two nucleotides, primer plus three nucleotides, primer plus four. And it goes on like this. Okay. So what it means is the dideoxynucleotides will be ending at each and every base pair. So wherever the dideoxynucleotide ends, you will be able to know what that nucleotide is. That is the principle of Sanger sequence. And you can see this second image, second work. These different sized fragments are run through this capillary electrophoresis and read at the last. So the smallest fragment and the longest fragment. Okay, and that's how we read it from here. So the sequence goes like this. After the primer, we have got T, TP, G, T, C, C, and that's how it goes on. Next is the applications of Sanger sequencing. Sanger sequencing has been instrumental in numerous areas of research, including genomic analysis, molecular diagnostics, and even in forensic identification. And Sanger sequencing was the key tool in the whole genome project, human genome project. It has been used to sequence the entire genome, and sometimes it is used to identify the disease-causing mutations, genetic variations within populations. These are some of the applications of Sanger sequencing. It has been used in biology and microbiology, in so many other fields as well. Coming to the conclusion, the Sanger sequencing has truly revolutionized the human genetics research and genomics research, and even in microbiology and virology, in all the fields, it has revolutionized because coming to the cost, doing a whole genome sequence for identifying one gene or one gene fragment is too much. Whereas Sanger sequencing is very cheap and it is very helpful to identify a small region of a gene or the 16S RNA or any viral sequences. It becomes very handy and truly it has revolutionized the genomics and genetic research. Thank you, everyone. If you have any doubt, you can write to this email, or if you have any feedbacks, you can write to this email. Thanks for watching my lecture. Thank you very much.